Hello friends, today we are going to discuss one of the important topic about DNA methylation and its role in gene expression. DNA methylation and its role in gene expression. In last class we have discussed that tiny chemical modifications in the chromatin structure have a huge bearing on gene regulation. For example, histone acetylation. Say lysine residues has been subjected to acetylation and will open up the DNA. That means the loosening or uncoiling of DNA that leads to gene transcription. That means expression is on. But deacetylation, expression is off. Just addition of or deletion of the acetyl group COCH3, which had a huge impact on entire gene expression, in turn of course entire life or cellular machinery. Under the same head we have DNA methylation. That means addition of methyl groups to the specific DNA sequences also had an impact on gene expression. In general it is said that methylated DNA leads to inactivity of genes. That means expression is off, there is no expression, because the methylation which generally takes place in CG sequences, what we call, that means the cytosine nitrogen base of CG sequences has been subjected to DNA methylation at the 5th carbon, what we call 5' methyl cytosine, and formation of this methylated DNA prevents transcription. How? Because methylated DNA binding proteins will come and bind to these methylated forms, therefore which will not allow the transcriptional elements to come and participate, that not proceed the gene expression, because methylated form, that methyl group projected out of the major groups, therefore become easy liable to the binding proteins. That means methyl DNA, methyl cytosine binding proteins, which will block the DNA. There is no transcription. So that what we call inactivity of genes. In simple sense, methylated DNA leads to inactivation of genes. Demethylated DNA leads to activation of genes. Just like in yesterday's class, histone acetylation leads to gene expression. That means expression is on, whereas histone deacetylation leads to gene expression off. The same concept, but here methylation leads to inactivity, that means expression is off, whereas demethylation leads to expression is on. That's the basic concept. Okay, and as we said, DNA methylation takes place in cytosine of CG sequences. Its rate is like majority of CG sequences, around 20% are methylated. Majority of CG sequences are methylated, and I said that it is at the 1.5% of genome reported to have 5 methyl cytosine. See, 5 methyl cytosine. That means its rate of occurrence 1.5% of, or 5 methyl cytosine occurs at the rate of 1.5% of genome. Its rate of occurrence is 1.5%. And CG sequences are the hotspots. CG sequences are the hotspots for DNA methylation. This is very important. And which enzyme? Just like yesterday, there should be an enzyme, what we call DNMT. DNA methyl transferases. DNMT. There are 3 families of DNMT responsible for this process. DNMT1, DNMT3A, DNMT3B. This is very important. So in simple sense, methylation leads to inactivity of genes. Inactivity of genes because of binding of methyl cytosine binding proteins. So gene expression is off. These are the basic issues. So since here gene expression is off, there is no activity. Since here, because why? I told you, blocking. Say for example, if it is open gene expression, the methylated, that means the proteins which binds to methyl cytosine will come and bind. That means blocking the DNA, which will not allow the transcription elements to come and bind. Just like in histone acetylation, loosening the DNA structure will enable the transcriptional elements like RNA polymerase, transcriptional factors, so that they can proceed with gene expression. But here it won't, because they completely block. The same concept, but the kind of chemical, whether it is promoting or repression. And most interestingly, DNA methylation is correlated with histone deacetylation. Histone deacetylation. This is very important. That leads to double gene repression. Double gene repression. I told already. Histone deacetylation means it will again recoil. Here, uncoiling is done because of histone acetylation, because of addition of these acetyl groups which bind to the lysine residues present in the N-terminal tails of histones. But deacetylation again coiling. Surprisingly, that means actually interestingly, these two are interconnected, because they both are working on inactivity of gene expression, what we call double gene repression. I told you there are two variants. If one is promoting, the other will be suppressing, that means repression. In case of histone acetylation, promoting the gene expression, deacetylation repressal. It's the same concept. DNA methylation is repressing, whereas DNA demethylation will be promoting. What we call epigenetic gene reprogramming. What is that? Actually, all this, particularly the DNA methylation, shall be studied under the broad head of epigenetics. Epigenetics. What is epigenetics? It is nothing but the study of heritable changes in the DNA. Say DNA methylation is a major point, but without changing the sequence of DNA. Here there is no change in the DNA sequence, but there are changes in the DNA. That means chromatin will be subjected to change, and it has been heritable. And the change is heritable change. Say the chemical modification, DNA methylation, it's a heritable change. Therefore, it's had a huge bearing on cell proliferation during embryonic development. What we call epigenetic changes, and DNA methylation is a major important process. How this process done, we'll discuss point by point. Say this is the normal cytosine. Oxy 4-amino pyrimidine. Okay, this is a normal cytosine, and this is DNA. Okay, then whereas its method form, what we call 5-methyl cytosine. Simply at 5th position there will be a methyl group, that's it. Two oxy 4-amino pyrimidine. Here methyl group. This is the methyl. 5th place, no? This is 5th. This is 1, this is 2, this is 3, this is 4, this is 5 and 6. 1, 2, 3, 4, 5, 6. No? It's like clock. The pyrimidine is clockwise. So the addition of 5th, that means 5-methyl cytosine. The methyl group at the 5th carbon become 5-methyl cytosine. And who is responsible for the DNMT? DNA methyl transferase. This is very important. 1, 3A and 3B. Okay, I will tell you how it. And how they prevent the gene expression. This is very important. Say for example, I told you already, CG sequences where the what we call DNA methylation will take place here. Say consider a gene, a particular gene, and these are the CG sequences. This implied CG sequences. Okay. Here there is no methylation. DNA binding proteins. That means here the start of either the start of gene, that means RNA transcription. Okay. That leads to gene expression. The DNA binding proteins will come and they will start. Say here the RNA will set, the RNA polymerase will set, then proceeds the gene expression is on. Here there is no methylation, no methyl group. But in another variant, take this is A and take example as B. There is a DNA methylation. I told you already, these are these CG sequences where will be subjected to methyl attack. Methyl attacks. Then we add the CG sequences like this. That means the methyl groups CH3, 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 and this leads to blocking the blocking the genes. Just the block. For example, I am saying blocking by what we call methyl cytosine binding proteins. Methyl cytosine binding proteins. Here they come out of major groove. They project out of, that means project out of major groove. Then that leads to gene expression of. Gene expression of. So there are two classical. It is take classical example. Here is a normal gen strand, for example, and there are CG sequences, and there is no methylation. We all knew that. Say the RNA transcription will come. That means RNA polymerase will come and allow the transcription like mRNA and all that gene expression. Okay, but here there is a methylation. That means that through DNA methyltransferase, it will add the methyl groups at the 5' ends. Then here the DNA binding what we call methyl cytosine binding proteins. Methyl cytosine binding proteins is very important. These methyl cytosine binding proteins will block. They bind and block, and there is no transcription. That means they will close off the gene so that the transcriptional elements shall not participate. That leads to gene expression off. That is a typical role of DNA methylation. Okay, this is fine. How the expression and how the mechanism I have discussed about. I told you already. DNA methyltransferase, DNMT 1, DNMT 3A, and 3B are responsible for methylation. And how about demethylation? DNA demethylation. This is very important point. DNA demethylation, it is just like histone acetylation. It has to promote the gene expression. That means gene expression is off, and who will take care of it? Just like DNA methyltransferase, there is another. It's a complicated process with respect to DNA demethylation. I'm saying a group of elements in a sequential step will be done. Say TET, what we call 10-11 translocation. That means TET is an oxidation mediated enzyme. That means TET belongs to 5-methylcytosine hydroxylase family. This will mediate the demethylation not in one step but in a sequential step. That means addition of methyl group to the cytosine, that means forming 5-methylcytosine, will be easy, but demethylation is complicated because we have to sequentially remove the demethylation process. There are some certain intermediaries will form during this sequential process, what we call modifications of cytosine bases along the chain, that means along the cycle of demethylation, and different elements will come. And this is very important. I told you why, because demethylation is important role in epigenetic gene reprogramming, because the gene has to again restore, it has to again switch on. For that, this has to be done in a sequential process by involving a set of enzymes, and I will tell you how it will be done. Okay, so this is 5-methylcytosine. I'm not drawing against the structure again because I already drawn. This is 5-methylcytosine. Okay, then it comes from normal cytosine, we have discussed already. How? By means of an enzyme what we call DNA methyl transferase. Okay, this is normal. And we have to discuss about demethylation. How? This 5-MC, what we call 5-MC, 5-methylcytosine, will convert first into 5-HMC by means of the hydroxyl, means by means of TET. Here TET stands 10-11 translocation, and here TET are three different kinds: TET 1, 2 and 3. All these three involving in DNA methylation. How? The first intermediate 5-HMC will form, later the 5-FC will form. Again here TET. That means TET1 here from 5-methylcytosine to 5-HMC, and TET2 5-HMC to 5-FC. Then one more, what we call 5-carboxyl cytosine, 5-FC, carboxyl. Here FC means formyl. Here, that means entire chemical modifications taking place as 5th carbon. First the methylcytosine, then 5-HMC, then 5-FC, then formyl cytosine, then 5-carboxyl cytosine, again by TET. This is very important. And this 5-carboxyl cytosine will become, that means normal cytosine, what we call the last step of demethylation. But here the not the TET, that means now the 10-11 translocation of hydroxylase enzyme, it is not that, but there is another enzyme, what we call, this is a different process. Thymine DNA glycosylase. This is an enzyme responsible here first. Thymine DNA glycosylase. Base excision repair. Base excision repair. Sometimes 5-FC directly converts into cytosine by same process, TDG and PER. This is how the DNA demethylation cycle will run. You know, the thing is the same, the concept is simple. In one step methylation will be done at 5th carbon, but not the DNA demethylation, because in a sequence, what we call a sequential cycle of some cytosine base changes by involving set of complexes enzymes, say TET1, 2, 3 and etc. Other, I will tell you first of TET1, 10-11 translocation 1 from 5-methylcytosine to 5-HMC, then 5-HMC to 5-FC by another TET2, then 5-FC to 5-carboxyl cytosine, another TET3, that means then the final step. This is very important. Actually, we have studied in our previous classes about DNA damage repair mechanism, kind of DNA damages and how it get repaired. Here, when there is a mismatch, there will be base excision repair, what we call BER. Simply it will cut down the unwanted base and will include the healthy base. For another example is double strand breaks, DSBs. This is how the issue ends. With respect to DSBs, we have non-homologous end joining, NHEJs. Okay, the same concept here. Here, a wrong base has to get transferred by means of BER, base accession repair. That's it. And then before that, thymine DNA glycosylate. Then after that, BER, base accession repair will come into operative. So that will convert an what we call a unwanted base into, that means a wrong base into normal cytosine. And sometimes because of this occurrence intensity, I told you already, 1.5% of genome reported to have 5-methyl cytosine. This is informally considered as a fifth base. Fifth base. 5-methyl cytosine is considered as a fifth base. That is how the issue ends. 5-HMC to take 5FC, then 5-carboxyl cytosine, then the cytosine by TDG. Thymine DNA glycosylate. But sometimes directly without 5-carboxyl cytosine, the 5FC directly converts to cytosine by same process. Thymine DNA glycosylate and BER, base accession repair. This is how the DNA demethylation takes place. I told you it's a bit a complicated process with respect to DNA demethylation, but it is very important in respect to gene expression, because I told you epigenetic gene reprogramming. Again, the gene has to come back to its own normal position, and want to remove the unwanted activity of DNA methyltransferases. That means preventing the entry of DNA methyltransferase is most important. For that, this DNA demethylation is essential. This is how these two, what we call coupling process, methylation and DNA methylation, come into play and has a huge bearing on the chromatin structure. I told you already, these are under epigenetic study. That means it leads to, say, after cell division, these DNA methylation become heritable. That means they had a huge role on cell proliferation development. Okay, so about how the DNA methylation and its role in gene expression. Okay, and thanks for watching. We will discuss an important topic in some